Okay, hello, happy February. So today we're getting into Osteichthyes. I just told you guys, like when I the first time I taught this class, actually, this lecture was called A Bunch More Fishes, because I know it's just been all fishes almost all the time when we've been talking about vertebrates. And so that paraphyletic assemblage of vertebrates that swim around, we're gonna bust free a little bit of that today with these bony fishes. And so I like this paleo art really very, very, very much. Not only is it showing you a lot of biodiversity, if you squint and look around there, you can see different kinds of fishes. There's sarcopterygian fishes, tetrapods, sarcopterygian fishes, actinopterygian fishes, a coelacanth lurking in the back. And it's all in this like flooded, forested, freshwater environment. There's a lot of information here that's empirically grounded to help us understand not only bony fish evolution, but also our very specialized kind of lobe-finned fish, tetrapods, fishes with arms and legs, kind of showing up. This is that ecological situation, and we're going to get into it today. But we have to start a little farther back. Here we are, just to get you guys settled in, as usual, to vert paleo. So here's our diversity of the modern world today. You've seen this now so many times. So our vertebrata node with cyclostomes on one side and jawed and gnathostomes on the other side. I hope I overdid it by telling you all these things about pieces of information, data we can use to understand jaw evolution. I think it's super cool that a cartilaginous jaw arch in some things had bone from the skin wrap around it, and some of those scales became things that are now teeth. And so you have this structure that's not actually itself, this bone is not a gill, but it used to wrap around a cartilaginous thing that was a gill. I think jaw evolution is so cool. And I am very interested to hear what you guys thought about last Tuesday. And if it was like, Brandon, that was just too much stuff. But now you know where jaws come from, and I feel like I've done my due diligence. So once we're in jawed fishes, jawed vertebrates, you get your bony fish and your cartilaginous fish. You guys learned a little bit, a couple characters, the prismatic calcified cartilage and the claspers that unite all the living chondrichthyans, the cartilaginous fishes. Today, though, we're taking the left and going into the bony fishes, where the absolute lion's share of vertebrate diversity is today. Most vertebrates today are Osteichthyans. And so within Osteichthyans, you have your lobe-finned fishes, sarcopterygians, and your ray-finned fishes, your actinopterygians. If you add them all up, include all the living tetrapods, there are more sarcopterygians today than actinopterygians. But actinopterygians alone are nearly half of vertebrate diversity today, ray-finned, bony fishes. Okay, here is that on the phylogeny with a paleo bend, same Snapomorphes mapped, same nodes labeled. But now looking at the fossil record, the stratigraphic record, time, as well as phylogeny, and including cool extinct fishes that help us understand that evolution. What I want you guys to do now, work with the people around you. Just as a refresher, here's these different synapomorphies that we've gotten to get up into gnathostomes. There's 2 for all gnathostome-line vertebrates. There's 3 that unite the osteostracans with the gnathostomes. There's 3 for all jawed fishes. And there's 1 that some placoderms have that all of us gnathostomes inherited. So go ahead and talk to your neighbors. What are these? Make sure we're fresh. Oh, yeah, bone scale. Oh, that's the big revelation.
Brain case.
Would it be the
consider people coming next school, but it's also you know fighting for
Okay.
oh wait, no, John.
I started.
Okay.
All right, so there's 4 little categories here, 4 little bullet points. Any group want to tell us what they think, what they're recollecting?
For the first one?
The first one, meaning the 2 that all Nathostoma line vertebrates have, all this over here.
I'm an idiot. Never mind.
You are not an idiot. What did you mean, this one?
Is I was branching left and right and, just totally missed that we were talking about only jaw.
No, well, this is the jawed line. I'm asking you guys, what are these? Because I just throw these phylogenies at you and put red tick marks. I'm just trying to make sure we are grounded a little bit. Okay, another, Jonathan, you're welcome to tell us, but.
Well, my first thought was, you know, jawed and jawless, but, I don't, right.
So where did you wear a jaws?
The jaws,
Which one of the red tick marks is jaws?
the jaws would be on the Nathanstones.
So if I was gonna put it 1, 2, 3, or 4, like where did the jaws go?
It would be on the far right.
The far right, this one?
Yeah.
Agree, disagree? People are disagreeing, sorry to say. So okay, so who wants to tell us, what's this one? Teeth.
Okay, so it would be before.
This is
that's-
teeth.
that's what's-
So some of the placoderms have teeth like you do, and all these features are teeth. So 1 that are shared by placoderms and all today's Nathostomes, for sure that's teeth. All right, other groups, what are these ones? Yeah, Gary.
The first 2.
The first 2, so everybody that's on the jawed vertebrate side of things, not the lamprey hagfish side of things, what are these 2?
The dermal skeleton and the lateral line system.
So a dermal skeleton, some sort of like almost an exoskeleton, an armory bony thing, scaly thing, and a lateral line. So okay, that's there. And then here's this complicated myth. What are these 3? 2 sets of 3.
The Osteichthyes Nathostomata.
Yeah, so these are little cuties that don't have jaws, plus everybody else. These 3 synapomorphies for that clade are
Dorsal fin.
dorsal fin,
Pectoral fin.
pectoral fins,
And then heterocircle.
heterocircle, tail.
Yeah.
The tail. So that means the vertebrae go up into the top of the tail. You guys met in aspens a long time ago, and you're going to meet other vertebrates later where their vertebrae go down to the bottom part of their tail. It's still a vertebrate tail, but it's structured differently. That's called a hypocircle tail. So heterocircle tail, pectoral fins, and dorsal fins, all finny features. Okay, and then what are our 3 synapomorphies that unite all the jawed vertebrates, extinct or still alive? What are we talking about?
Pelvic fins.
Pelvic fins. So now we got these. Okay. 3 semicircular canals in your inner ear, and thank you, jaws. So here's those characters. When you guys are like kind of like studying and stuff, and I'm gonna give you your first of these like take-home homeworky things this week. This is kind of what I'm hoping you're able to do as you're like reviewing our material, re-looking at lecture slides, understanding how these features, which might be some of them important to you and your body right now, some of them were in your ancestors and you have lost, how those are distributed here, how these synapomorphies help us put together all this biodiversity. Thank you for humoring me. That was about the lowest common denominator, kind of asking you guys a question. It was just like, hey, I already told you this, which one's which? But thank you. It's just, I hope forces you to think and put these things on this tree in your head. Okay, now let's go forward. Right at the end of the class on Tuesday, I gave you 2 more synapomorphies that unite the living chondrichthyes, cartilaginous fishes. That was the claspers that the males have on their pelvic fins. And you guys now know pelvic fins are a Nathostomata thing. And then also the way they do kind of mineralize their cartilage. They have prismatic calcified cartilage. There's little cartilaginous skeletons in a lot of these animals on their vertebrae, but sharks and rays, and today they actually do mineralize in a certain way. They have a cartilaginous skeleton, but it's a special kind, prismatic calcified cartilage. All right, that's those living fishes that are cartilaginous. The rest of the day today, we're going to this side of Nathostomata, to the bony fishes. So that's you and me and coelacanths and lungfish, plus all those ray fin fishes, the Osteichthyes. I've got three characters that unite, three synapomorphies that unite Osteichthyes that I hope you guys can take with you. Finally, it's February. You guys are in vertebrate paleontology, and now that we're in February, all of January has passed, and we finally have what we call a bony internal skeleton, a fossil record that has most of the bones of the body. So when I say a bony internal skeleton, I'm talking about these endochondral bones that come almost all from that mesoderm cell layer when you're an embryo. They're preformed in cartilage and then turned into bone as the animal grows up through its life. I showed you this already. This is like a little neonatal care pamphlet image that I think is amazing for new moms and dads, telling about how their baby's bones are forming. Here's an image, just so you know, I'm about to show you a medical specimen. Image of a little human embryo stained. And so you can see how almost all the bones of the skeleton down here, the endochondral bones, form in cartilage. And then starting at the center, there's a collar of bone that grows out. And so as a baby grows up, as you grew up, your wrist bones and your fingers all ossify, and they come from the center of each different bone structure. Your skull is completely different. And I hope the mind-blowing thing you guys got from the last week and a half is that these dermal bones, especially ones like the maxilla and the dentary, are homologous to armored placoderm faces from back in the Paleozoic, and they form out in the skin. It's from an ectoderm cell layer. So this image that I love so much that hospitals might give out to you if you have a baby, if you squint and imagine you're looking at it like this, that's like a little placoderm, armor, and then a cartilage endoskeleton, but it's a human. And I just think that is the coolest. And so we see that in all Osteichthyes, not just us tetrapods. So here's a fossil of a ray-finned bony fish. And so there are endochondral axial, which means the spinal column, the axial skeleton, and appendicular, which means the limbs or the fins. Endochondral elements, so preformed in cartilage. We can look at their histology and see that. We can watch it in living ray-finned fishes all over this fish's skeleton, just like all over this of a human skeleton. But then the cranial bones, most of them, especially the ones on the outside, things like the maxilla, things like the frontal, they're formed in an intramembranous cell layer. They're dermal. And so the fishy skull and your skull versus the fishy spine and your spine forming in the same way. This is a shared Osteichthyes feature, these endochondral internal bones. So finally, bones is what I guess I'm trying to say. So bone, vertebrate bone, is a really, really, really incredible tissue. I think most people think of bone as this kind of rigid support structure that all your cool muscles and nerves and blood vessels have to attach to or move past. Bone itself, though, is absolutely very, very alive. It is not like enamel, which is like a crystalline structure that breaks and that's it. It's just a crystal. Bone is living and always growing and changing. We're going to learn a little bit more about this later. This is a cross-section of a femur bone in you. Two different medical textbooks. I just like them because they both show slightly different things. And it's a reminder to you guys that your bone is a living thing. There's nerve cells, there's nerves, blood vessels, nutrients and waste coming in and out. There's cells that are forever living inside your bone tissue that are trapped, that have these teeny little passageways so they can still talk to each other. But when you break your bone, those cells are then what activate and then can put down more crystalline structure. Your bone is very much alive. It's a really highly derived tissue. Only Osteichthyes have this endochondral bone that has this ability to really, really, really drastically keep itself healthy and remodel and do all these things. Some of you might know that on the inside of your marrow cavities in your bone is where things like red blood cells are made. So these are organs in your body that just happen to also be physically the structure that you rely on. And so bone, like this, is an Osteichthyes feature. So there's one of those little living cells zoomed in. So if you've never seen something like this before, there's these little cells living all throughout your bone that have these teeny little connections between each other so they can talk. Later in class, we're going to talk about histology. How can we learn how fast a dinosaur grew? We can cut into the bone and look at these cellular structures to get after those questions. It's only possible in bone like this. Very, very cool. Okay, something that's related to that. So the first synapomorphy for Osteichthyes is that bony internal skeleton. Bony internal skeleton. There's three I have for you guys for Osteichthyes. The second is related to a bony internal skeleton, but it has to do with the dermal bones, the outside bones, the bones that are homologous with some of that placoderm armor. Dermal bones are the ones that do support teeth. And so here's a ray-finned fish today, a ray-finned bony fish, an actinopterygian called a gar. And here's a nice iguana that, whether you want to accept what I'm about to say or not, has a skull a lot like yours in a lot of ways. Both of these Osteichthyes have homologously shared from a common ancestor teeth on the premaxilla, teeth on the maxilla, teeth on the dentary. This is an ancient bony vertebrate thing, to have those teethy structures put on those dermal bones. So that's something I want you guys to have. We're going to talk a lot more in lab next week about skulls and skull bones. If you don't know some of these words, we're going to do that in lab next week. You'll have a lot of specimens to play with. And then finally, a pretty surprising Osteichthyes feature that has nothing to do with bone. It has to do with the rest of their bodies. This is called a foregut diverticulum. And I'll let you take the time to write that down. I know it sounds kind of awful compared to the ones I usually give you. And so what you're seeing here is an ontogeny of part of the foregut. So the gut is the tube, that endoderm line tube that goes from your mouth to your anus. It's your digestive tract through your body. The foregut is the beginning of that. And in the front part of Osteichthyes, you get, as the embryo is growing, a diversion off of that gut that forms these little pouches. And those pouches are ancient. All Osteichthyes share those pouches. Let me show you what they look like. Here's a frog. You're looking at the frog's gut passing through here. Its foregut is the front. So here's the mouth, here's the throat, here's the frog's stomach. This is that passageway in a frog. In a frog, it leads to its lungs. So a frog can take in air. And then in its lungs, it has these specialized tissues that I'm sure you guys already know about that exchange oxygen with the air, a lot of blood vessels. So that frog is breathing with its lungs. It has a little glottis, so a little muscular closure to make sure food and water doesn't go into its lungs. But you guys, I'm sure, have heard about the Heimlich maneuver. And you're aware that us humans and all of us tetrapods have this kind of design problem where our lungs are attached to our throat. So your lungs are an offshoot of your digestive tract, which is very cool. And so they are connected right here. In frogs, we call those diversions, these highly derived features called lungs that are breathed with. In a fish, like a carp or a minnow, we can use as an example, we still have a mouth, we still have a gut, a throat that goes to a stomach and then intestines and out the back. No one's surprised. But a fish, people have known for a really long time, fish doesn't breathe air. It breathes with its gills, which is a totally different thing. But it still has this little diversion up here, this ancient diversion that's shared with the tetrapods. And in a fish like a minnow, it goes to this gaseous balloon structure that I think you'll agree looks a lot like a lung. And in most fishes, that's called a swim bladder. Lungs in a frog, swim bladder or gas bladder in a fish. Fishes can put gas into that from their blood. It's like a reverse lung. They can take dissolved oxygen in their blood and put it into the swim bladder, fill that swim bladder with air, and they use it for buoyancy. Highly evolved ray-finned fishes use this structure to move around in the water column. A frog uses this structure, and so do you, to breathe. The point I want to make to you is this diversion, foregut, diverticulum, is very, very ancient. We can look at embryology to see that it's the exact same structure and forms in the same way in these different animals. What is extremely cool, and again, paleo can come in and help with this conversation, is that the common ancestor of all Osteichthyes had gills, of course, that we assume it's using to oxygenate its blood, but it also had what we could call lungs. The thing that you see in most of the fishes today that you would catch on a hook and is called a swim bladder, that is probably a super specialized thing that used to probably be able to function as a lung. Other Osteichthyes, the ones that are close to us, like lungfish, are famous because they can come up to the water surface if they're in a pond that has bad oxygen levels, low oxygen levels, and they can go, and they can use that diverticulum, put air into their lungs, get oxygen out of that air. Everybody kind of knew that. Something that's been really coming out in the last 20 or 30 years as people get really detailed about it is most of these ray-finned fishes, especially the earlier diverging lineages, so not like a bass or like a cute, Nemo fish, but like a lot of these early fishes you're going to meet that are ray-finned fishes, they all functionally can, if there's low oxygen conditions, go up to the surface and gasp some air and oxygenate their blood with their swim bladder, which is the same thing as our lungs. So what's really, really, really crazy to think about is, based on the modern diversity, it lets us be like, wait a minute. Can all Osteichthyes straight up breathe air if they want to, as well as already have their gills up front? Very, very cool. And so we're going to talk more about that today. Here's those two features that you've already seen on the paleo tree for chondrichthyans, prismatic calcified cartilage, and claspers. And now here's those three features I just gave you for Osteichthyans: a bony internal skeleton, teeth on the dermal bones, and a foregut diverticulum, AKA lungs, is this ancient. So think about how crazy that is. We know that you can breathe with your lungs and you can breathe with your lungs. I wonder what this fish is doing. And if we go this way, you can breathe with your lungs. There's other lineages that can breathe with their lungs. You guys really don't, but you have that structure and you use it for buoyancy instead. You've adapted it to be something else. That's something that's really challenging, because I think in most people's minds, they think like shark, fish, amphibians, and blah, blah, blah. So when we talk about fishes moving onto land, it feels like all the specialization is over here. And in fact, some of it's more ancient, which is really cool. All right, there's our three here. We have our Osteichthyans, and on one side is Actinops, the ray fin fishes. On the other side is Sarcopterygians, the lobe-finned fishes. We're going to talk about Actinopterygians first, because I don't have as much to say about them as I do about Sarcops today. So here's our Actinopterygian biodiversity slide. These are our ray-finned bony fishes. I'm showing you art and one fossil, I guess, of Devonian Actinopterygians. These are the most diverse single group of vertebrates today. There's over 30,000 species of Actinopterygians still alive right now. In the Devonian, they're not very big. They're round, they're small, they're in marine environments. There's a lot of them in freshwater environments, which is very interesting, especially if we know that they probably could breathe air if they wanted to, probably. Something that I think is really cool is Actinopterygians show up in the Silurian. They're diverse in the Devonian, but small. But even the living Actinopterygians that we have today, the oldest divergences amongst them also are in the Devonian. So there's lineages of Actinops today that go all the way back by themselves to the Devonian. So I'll show you what I mean by that. Here's a phylogeny of ray-finned fishes. There's this one, Moitamazia, which is only in the Devonian. But this split right here is the crown group of those ray-finned fishes, Actinopterygii. We'll meet them a little later in more detail, but this is called a bichir. This is the kind of fish that lives in Africa today. Guess what? It walks around on the bottom on the fins. And if it wants to, it can go to the surface and breathe air. And so it's more closely related to like a goldfish, but it still has all these things that are kind of interesting for you guys to think about. It can like walk around on the bottom and it can go at the surface if it wants to. That's interesting. The rest of the diversity of Actinopterygians we're going to see expand again and again and again as we move through time in this class, because there's, as you know, so many Actinopterygians. All right, what I really want to do today is take a left and spend a lot of our time in these lobe-finned fishes, these Sarcopterygians. And so I've got one character for you guys to write down, for you to remember about Sarcopterygians that I think is very cool. It's been known about for a very long time by anatomists. And this is called the monobasal. So what you're looking at here is a phylogeny. It's got time on the bottom. And what you're looking at are the pectoral fins of all these fishes. So here's our chondrichthyans. There's a shark's pectoral fin. Here's our Osteichthyans that split into Actinops. You can see some of the Actinopterygian pectoral fins. Here's our Sarcopterygians. You, lungfish, and coelacanths. I'm saying you, but that's a frog's arm. Okay, talk to your neighbors for a second, just to like break up the fact that I've been blabbing at you. What are you observing about these fins relative to each other? Pectoral fins.
Yeah, it is like a separate goal.
I'm sure there was some risks.
Yeah. You know, you will. Yeah. All that. Don't just like this, don't want it.
Everything up there. Oh, not so much bigger. Variation of day.
There's a lot of variation.
Yeah, that doesn't bad further.
Yeah.
It's really like, what is this?
Like the others.
Okay, so they're all modified on it, they all.
Yeah. The fingers are not.
Yeah, so what are some general things we're talking about? I'm hearing good stuff. Yeah.
The one which sort of has fingers, but they're arranged very differently.
Serially, if I had like fingers like this. Okay, so there's little bony spurs off the lungfish fin, right? Interesting. I think these are interesting for you guys to look at too, because I bet when you think of a fin, you just think of like the big shape that it is, not the little detailed architecture that's inside. And so the shark actually has all these structures that look familiar maybe, or look similar to these other ones, but then shark, it's all cartilaginous, which is kind of cool. Anybody talking about the Sarcopterygians and how they look different? Because we're on like literally a slide that's about the Sarcopterygians synapomorphies. Anything jumping out about like what seems different about them?
To me, it looks like if there's like one sort of central bone, but in charge, or
Leaning into that word mono right at the top. There's got to be something with one, isn't there? It's like a unibasal, a monobasal. Mono means one. And then that's totally right. So the basal, if you take in like this fish, which is a gar, or you guys probably know what like a sturgeon is, in the base of a pectoral fin here, these little bones are called basals. And there's a few of them in most kinds of fishes, including sharks. Even if they're made out of cartilage, they still have the same homologous like structure. In these Sarcopterygians, there's only one basal that actually articulates with what we call the shoulder girdle or the gill skeleton of these fishes, a monobasal. What is really cool about that monobasal in that coelacanth, that monobasal in that lungfish, and this thing in a frog, you guys know what that's called in a frog. It's a humerus. The monobasal is literally the humerus. So a coelacanth and a lungfish have a humerus like you have, and it's the only bone in their pectoral fin that actually touches, guess what they also have? Their scapula. And there are many other bones that they might have, a clavicle, a clitorum, there's a whole bunch in their gill skeleton. They still have a little humerus. What's cool is we can do developmental biology, study how the tissues evolve, grow in an organism, like through its lifetime. And a coelacanth has a radius and an ulna too. And so does the lungfish, but they're fused. That's a fused radius and ulna, but they form separately and fuse as the animal grows up. So what's cool is like a little one of these is in these fishes. Really fun. And so the development of our limbs from fins couldn't be a more interesting and popular paleontology topic, because we can look at the fossils and be like, well, what happens? What are the details? So monobasal is the feature I want you guys to have that unites all Sarcopterygians, coelacanths, lungfish, and us tetrapods. Here's my Sarcopterygian diversity slide. They also show up in the Silurian. We have good fossils. Some of the first Gnathostome fossils I showed you guys last week were Sarcopterygian gnathostomes all the way back in the Silurian. So remember I showed you like Megamastax and Squarolithus, some of these fragmentary fossils. But in the Devonian, they get very, very diverse. There's three big groups. On this slide, I'm showing you a fossil lungfish that's from the Devonian, a reconstruction of an early coelacanth, also from the Devonian, and it kind of looks very similar to like the coelacanths that are absolutely alive right now. And then all four of these down here, three pieces of paleo art and a cool fossil, these are all tetrapodomorphs. So these are fishes that are closer to you and me than they are to lungfish and coelacanths. So there's diversity even within Sarcopterygians, the lungfish and all their relatives, the coelacanths and all their relatives, us and all of our relatives. These four are tetrapodomorphs. These are fishes that are close to us, not even coelacanths or lungfishes. So a lot of diversity, marine and all that. Why don't you make some observations real quick? Talk to each other about these organisms because they're pretty awesome. What are you noticing about them?
Yeah.
Really,
Well, I do see it.
I think that's terrible. Their eyes are.
It's
Gotta be, I don't know, just people.
it's like.
They would probably.
Yeah, they got.
What are the things we're talking about? I'm here to look at those teeth. I like that. One thing that I think is really cool is if you got out of your time machine back in the Devonian, if you got out of your time machine like right here, out in the oceans, the really scary biggest predators are those placoderms like Osteichthyes, like huge bony skull mouths. In freshwater ecosystems, the biggest predators are tetrapodomorph fishes. So there's this little shark just getting crushed. Prismatic calcified cartilage is not helping. You're dead. So in lakes and rivers, this animal is like a 12-foot long fish. It's huge. Living in freshwater ecosystems, it's so funny to say this, our tetrapodomorph fish relatives are almost all the big predators, which is really interesting. So sarcopterygians do very well in the freshwater ecosystems. There are lungfishes and coelacanths out in the oceans too, but a lot of the sarcopterygians are in freshwater as opposed to all the sharks and placoderms out in the oceans, which is getting interesting. We're starting to spread it apart. Anybody else saying anything? I like this art because you can see the well-muscled pectoral fins. There's a little humerus in there. And then there's these pelvic fins that are kind of just trailing, but they're still sort of paired. Okay. Here's some Devonian diversity. Every single one of these fishes is a Devonian sarcopterygian. So there's a lot of them, and they can see they have very different body plans, very different things happening in their anatomy. Tell me about these fishes. Yeah, I'll actually do that too. So tell me about these fishes. If you look at them all, what strikes you?
Yeah.
You know, like, they look
Like some of them have maintained, you know, that tail. But then I see others,
very.
they look a little bit. Like the. It's still a tail, but it's not, yeah.
That was not crazy to us, so.
A lot of.
In a little more very.
Like similar to like.
All right, what are some observations? What are things you guys are wondering about? Yeah, Jonathan.
Is that, many of these fish have, like a tail still, you know, leading up into their fin, like going up?
Oh, you mean the vertebrae with the tail?
Yes.
Yes, it's a heterocircle tail, really clear in some of them.
And then in
So
other.
that's an ancient Osteichthyes plus nathostome thing that these fishes just still have. Okay, but some of them don't have it. Interesting.
Right.
Okay, what else? I heard a lot of good stuff. Somebody else can do it.
The positioning of pelvic and pectoral fins.
Say more, please.
Well, it's all over the place.
Yeah, this fish has pectoral fins way up here. And they're small. I mean, look how long this back, look how long this dude is. Pectoral fins up here, pelvic fins back here. I heard you guys talking about their heads, they have big heads.
Yeah.
They're almost all predators, that's for sure. What do you think this fish is doing versus this fish? Their ecologies are all really different. I'm not showing you body size here, they're not scaled. Some of them are huge, some of them are like normal fish size, you might say. You know, maybe a foot or two long. A lot of different ecologies. The thing that I think is so cool is when we sit here in the modern and you're like, I want to know how fishes came onto land because I care about tetrapods and I want to know about evolution. It really does you a favor to like mentally go back into the ecological and temporal context for when that happened and realize that amongst all these fishes, there's a species that's doing something of everything in these habitats. Crushing clams, eating prey, being big, being small, living in the ocean, living in the freshwater. Ecological diversity is pretty high. So you only need one of these fishes to kind of be like, you know what? And start flopping around on land. And there's so many of them. Which one is us? Which one's the closest to us of all of these? One of these fishes, of those 7, is the one that's closest to tetrapods on land right now. Which one is it?
Wow. That's a hard,
Almost both like,
what? I haven't seen it. We'll see.
Similar. And you see the production.
All right, so which group has I know you're guessing. Which group has a guess?
Has a guess or?
Yeah, Jacqueline, what do you think?
I don't know if it's one of the two.
I definitely didn't label them on purpose, so if you were like, well, this one's called this one, I'd be like, wow, yeah, okay, which one?
The tan one and the.
Tan one?
Yeah, those two that branched off, I
Uh-huh.
would say probably my guess.
One of these two?
They're low fins or really low.
Low fins or really low-beat? Okay. Anybody have another guess or agree? Yeah, Stuart? What's your group talking about?
Far right.
The far right, this one?
Yeah.
Any
His
particular
front
reason?
stands look very strong.
Ooh.
Like, you could call him like.
Swiping. He looks strong. He's got good fins. Okay, what else? Other suggestions? We've got this node here, one of these two. That's one group said. Maybe this individual fish here. Any other ideas? Seem reasonable? No one's feeling it was in one of the other ones? Well, I love it. Okay, so here's some diversity for you. You don't have to know these words. This clade's called actinista. That's the living coelacanth clade. So these are the coelacanths right here. And Rhipidisty is these two. So there's a monophyletic group that's this one plus this one. These are dipnomorphs. These are the lung fishes as they look to the Devonian. These three are all tetrapodomorphs. But guess what? That's the answer.
There you go.
So this is a very scary stem coelacanth. There's a bunch of these coelacanths that are scary predators with these raptorial teeth that stick forward. Awesome. I'm not going to talk about them. Here's a shell crushing, like bivalve eating, brachipod eating lungfish that like only cracks shells. Fantastic. We're not going to talk about it. Here's our tetrapodomorphs. This one is actually the one that's closest to us. What I like about that, and why I'm so glad one of you picked this fish, because I think this one does look the most like a tetrapod, is to make the point that tetrapodomorph fishes a couple of times really flirted with like being up on land. The one that actually works, that we are from, is related to this fish. And I'm tricking you by showing you this one that's definitely still got all of its fins. But I think that's what's cool. There are other tetrapodomorph fishes that are like probably capable of being on land a little bit, having their eyes on top of their head because they're always at the surface. They don't have anything to do with us. I think that's cool, right? So if you got out of your time machine, you'd probably pick this one. It's not that one. Okay. So here they are arranged phylogenetically with a couple new friendly fishes I've added on here that are getting increasingly unfishy. Here I can throw these nodes on there. You guys know these. I want you to know Nathostomata, Osteichthyes, Sarcopterygiae. I gave you these two on the last slide. You don't have to know them, but that's where they go. And that's very cool to me. If you had a tetrapod focus, you could arrange everything we've been talking about. So you're just driving towards tetrapods. And that's how you could arrange your thing with your Actinopterygian and your Chondrichthyan right here. Okay, so when we talk about fishes going up onto land, there's a lot of stuff to think about. This is truly, I think there's probably like 6, I think. Like gold standard paleontology is amazing. Questions that people who work on biodiversity might have, like whales are mammals, but I don't get it because They don't have legs and all these other things. Whale evolution, bird evolution, we're gonna talk about a few of these. Tetrapod evolution, a lineage of fishes that becomes terrestrial immediately. People who are scientists, people who are really anti-science, this is something they want to talk about because they think evolution can't be true. How would this even work? So this is inherently extremely important and very cool. And the fossil record is literally where we could be like, yeah, time, place, and how. Here's our best ideas. This is the empirical data. So I want you guys to think about this bigger question. You can see Saturday morning cartoons your whole life with versions of this joke. What are the challenges? What can you imagine? As a biologist, sit there. We're going to have you guys talk to your group for at least like 5 minutes here. What are the challenges a fishy kind of organism is going to have adapting to living a terrestrial lifestyle? So I want to come up with a big list here. Go ahead and talk to yourselves, talk to your neighbors. We're going to spend about 5 minutes. What challenges face these organisms in this transition? How can that set our expectations of what we might find and see? Okay, go ahead and chat.
It first to be, there's,
Like, there aren't very famous. Not the bird.
well, we can already.
Like, what are they eating?
Their eggs. They're also better. They like all this machines.
We know there's
Well, I think you said it was.
Yeah, that's like. Some of
different.
We go.
They're not, we're getting nice. It's certainly something nice. And they're still turning. There's like.
No, you're not. I'm not.
I guess one of the smelling as well, smelling carrots. Oh,
Yeah, right. And yeah, you guys can.
There's still.
All right, I want to hear from like, I don't remember which ones I like. No, maybe that's okay. All right, let's hear from, I want to hear from all the groups. I want to get a big list going. What are some things you can just kind of shout them out?
What they're going to eat on land, you know,
So
it's gonna be kind of difficult to catch a boat.
what you eat,
Yeah.
also how you, these are all predators as far as we can tell. How do you catch it? When you're a fish, you live in water. And so you guys have probably seen when fishes eat, they can go like, and like the water, you can just sneak up behind a little scrim and when you open your mouth, it gets sucked in. But if you go behind a cockroach, it doesn't fly into your mouth. So how are you gonna catch it? What are you gonna eat? Even if you're eating little bugs. All right, what other things?
Reproduction,
How, tell me more, reproduction, how?
like how you find mate when you're on land.
Oh, okay.
And also when you do find mate, did you go back into the water or like? how
For what?
in there?
For what?
I guess if they're intermittent fertilizing, they don't have to like lay eggs in water, but I
Mhm.
mean, depending on the system you've got going on, I do bring that on land.
So everything, really, finding mates, fertilizing eggs. Or I'm gonna say, how about this, fertilizing embryos? Won't make any assumptions about what I say. And then like giving birth or laying eggs, having the baby. What does it change? What's possible, what's not possible? So this is a very fun one. Reproduction from start to finish. All these different things, it's just gonna be very different. All right, what else?
We were talking about sensory system.
Sensors, but tell me more.
So smelling, hearing, seeing, they all have to be.
Okay. You got touch, you got taste. Yeah, okay, so, all right, go ahead, tell me, I love it.
Well, like, what,
The physics above and below water are different, like hearing,
Totally.
hearing underwater and above water, you completely different.
If a whale is floating off of Maui and it's like sings its beautiful song hundreds of miles away because of water's properties, the whale can be like, oh, nice. But that same whale in the middle of this, like American Falls, we don't hear it. Air and water transmit sound differently. So, okay, so physics of, this is an excellent suggestion, by the way, I didn't mean to be silly about it. Physics of senses. Light is really different, huh? The best case scenario you can possibly imagine, I can see like a couple hundred meters in water. If I was in American Falls, I could look at mountains over there, I could look at mountains over there. So air and water allow light to travel really differently. Hearing is crazy different in terms of perceiving sounds, right? What about touch? What were you guys talking about with touch?
well, we didn't really talk about touch.
What did you talk about? What did you talk about? Like the
Well,
chemical ones. Taste and smell is both chemical. What did
You're
you
talking
talk about?
about land or land systems, the feel vibrations of the water.
Oh, yeah. You've been living a fishy life. All these Nathostone line vertebrates can like feel the water move around them. That does not work on it, right? It's not working in there. How about chemosensation? So that's like things like smell and taste. What's the difference? Or what? No, I don't know the difference. What's going to be a challenge for you?
I mean, one of the things with that we were talking about with senses is just there will have to be physical adaptation, you know?
Physical adaptation.
Because you perceive things one way in one type of environment and your ecology changes.
Yeah.
And for example, eyes, you know, the physical eye.
Yeah.
is really more sensitive to light, it's gonna be drying out, it's gonna be.
Oh, yeah. Drying out. How about drying out? Reproduction, there's something to do there with drying out. Living your life in general, there's gonna be something about drying out. How some of your senses work drying out. Anybody know the fancy word for drying out? Dehydration is one, that's like for your system. I'm gonna use this word. Desiccation, you can just dry out like your whole body, like all the tissues. So desiccation is a problem. You're not in water anymore. right? What are the things people talk about? Challenges.
Locomotion,
Locomotion, what's a locomotion problem?
no longer buoyant, we're no longer like in water.
The thing I say when people bring up that is always, hey guys, what happens if I put a whale on a beach? Right? If you live in water and you're neutrally buoyant, you can locomote, you can move in certain ways because you're neutrally buoyant. On land, there's suddenly gravity that you have to deal with in a very different way. So, okay, so locomotion has a lot of factors. Gary, what were you saying? One of them
And a lot of
was,
stuff, like holding yourself up basically against gravity, if you're no longer buoyant. Or difficult, yeah.
yeah, there's not gravity in the ocean, like something will sink in water, but gravity is very different on land versus when your body is maybe for the most part neutrally buoyant in water. What are other locomotion issues?
Muscles
What do you mean?
to adapt a lot stronger muscles on land.
So, yeah, so supporting yourself. It's not only the movement you're gonna be having to undergo, but like the structural support to do all that. I'll say support and movement. This is one of the most severe habitat changes I think you can imagine. It's fun, and we're gonna talk about it a lot in a few weeks, when animals go the other way and they live on land for millions of years, and then someone is just like, and goes back into the oceans. That's cool, because some of these things happen in reverse. But this changes all these things, maybe not what you eat exactly, but all of these things are true for every single lineage that changes. So the plants that moved up onto land, all the different arthropods and mollusks that move up onto land, they all have these exact same issues. And you guys can probably think about different ways those lineages adapted to live on land. Vertebrates are what we're going to talk about. These are great. Other ideas, other things people talked about. Off the wall stuff that I haven't thought of. Yeah, Zach.
You brought up the problem of UV light, so in the ocean, UV light dissipated, the further down you go on the surface, there's nothing to stop it.
I'm gonna put that in a bigger category that it is a part of. All of a sudden, it's like, oh gosh, you can get like burned by the sun in ways you didn't. If you just lived 6 feet underwater, at least you could be almost totally protected from most of the sun's light radiation. It's only light for you. Out on land, it can be a lot more. It's radiation for sure. That's related to desiccation, but it's a different thing physically and principally. Anything else? Yeah, sir.
We had a salt regulation.
Oh, tell me about that.
There's some animals have different chemistry in their urine that reflects, whether they, actually, I don't know specifically whether they would want more salt, less salt. So
One of the most advanced systems you have in your body that like regulates your homeostasis or your kidneys, keeping your salt levels at like a reasonable level. There's other things it's doing too, your kidneys, but keeping your salt and like just the other chemical factors in your body regulated is a big deal. If you lived in salt water versus fresh water, that's already a change, but now you're not living in water. You guys talk about dehydration. Dehydration is related to this, osmoregulation, having enough water to keep your salts moving around, flowing healthily. So it's very different. If you live in water, you don't really have to think about it probably too much. If you're out of water, yeah, that's a big deal. Other things. Some of these are really easy to talk about for fossils. Some of them really aren't easy to talk about for fossils. They're all probably still something to think about. Anything else? Yes,
How do you breathe? How do you go from gills to lungs?
Oh, yeah, how are you oxygenating your blood? We know these fishes have gills, and you guys might know you have. What's cool is this is a completely different structure than this. This does not happen. Gills don't become lungs. Gills are their own thing. Lungs are their own thing. So there's no arrow here. But what are those relationships? How do those things work? I hope one thing that you've seen is like the basal condition, the ancestral condition for every bony vertebrate. Doesn't matter if it's an ectopterygian or sarcopterygian, is both of these systems are present. You breathe with blood in your gills that are attached to your throat skeleton, a big Juberstone feature to have that gill pouch, and you can oxygenate your blood. But some of these Osteichthyes can also do it a different way, where they fill a little side chute of their guts with air, and then their blood can get air from that. You guys call that lungs. So some of these fishes, all of these fishes that you just saw, for sure have both of these systems at the same time, which is really cool. All right, here's some of the ones I think are the most paleontologically possible to talk about. Locomotion and support, fully agree. How you're breathing. Here's a bunch of other ideas I always think about. We're not going to talk too much about some of these. but you guys kind of came up with them on your own, which is just wonderful. So reproduction, how heat works. Terrestrial environments are crazy. It can be very cold or very hot in the same day. If you live in a pond or if you live in the ocean, your like baseline temperature of the environment around you almost never changes, or if it changes, it's over the course of months. Out in Pocatello in one day, it can be very, very cold or very, very hot. So heat capacity, how you're like regulating yourself, very different in water versus on land. So thank you guys for coming up with all those. That was really good. Let's take our quick 3 to 5 minute break right now, and we'll go to look at the real fossils we have to document this. If you want to, you can talk to your neighbors a little more while break. If things about this you didn't think about, other people maybe made you go, So let's take our quick break.
I think I have with it.
So let's come back here and take a look. What I'm going to do first, before we talk about the different synapomorphies that we can map in our phylogeny of tetrapodomorph fishes, is just show you guys literally some of the specimens. So here's the genus. Here's when it lived, time-wise. Here's a nice cartoon of its skeleton, or the outside of its body anyway, and a nice artistic reconstruction. So thinking about these things, I'm not going to put anything up on this slide right now. Thinking about these ideas, these fishes are related to each other. We're like, they're sequentially closer to us. This one's closer to you than this one, but they're both closer than that one. You get it. Talk to each other. Knowing this as our framework, come up with hypotheses, come up with predictions for what we should see in a fish that's adapting to live on land. What do you notice in the anatomy of these animals? Okay, talk to yourselves, talk to your neighbors.
And the shoulder bone.
Small house. See that? Oh, yeah, we do. Okay.
You met in the old place like 6 full.
I think
Yeah, so there's like.
I'm gonna add 2 more. I didn't put them on the same slide so that you could see them and they were large enough to look at. I'm gonna add 2 more and I'll go back and forth. You can keep having the same conversation you're already having. But these next 2 are the same thing. The one that's at the bottom is the closest to us in terms of its phylogenetic relationships. And then it goes all through all 5. Okay, sorry, keep talking.
Yeah, that's your man.
Showing really nice. Looks like
It's like exactly, why don't we?
Probably should answer, you know, like, they can use that
Yeah, here we go.
More leverage.
When we look at, you know, it's kind of tell that, like those lenses are just get the time.
Not generally.
Yeah.
Like over time.
And then after that, then.
Or just.
Like I said, like still looking at very, like, doesn't, it's just very, you know, very self. Yeah. The problem is.
All right. I've really enjoyed listening. You guys are talking about a lot of cool stuff. I think, so I'm not gonna have you like share out what you've been talking about because I like that you're talking about it, making observations about these things. What I think is really wonderful about things like fossils is if we go out there and find enough, they all come from a place and they come from a rock that has a certain age. All these fossils are from freshwater deposits from like a 10 or 20 million year window of time. They have features of their ears, features of their brains, features of their like microanatomy that we can use to like make a phylogeny that has nothing to do with like, say, their fins or their like eye placement, and then put those together to like make a hypothesis of how they're related. All this is just raw data. Think of it as raw data. When you see these fishes in pop culture, they're always being given to you as like a story. Here's where we come from. Here's where we come from. It's more fun, I think, to like be like, hey, look, this is all just raw data. And if we put it together, we should expect to see fishes that have to deal with moving on land. We should expect to see fishes that have to change how they're feeding and moving. And then we can like look in rocks all over the planet and be like, hey, look, it's kind of what we thought. Maybe the details are different, or maybe stuff's weird, but this is like matching ideas we could come up with. If you didn't know anything about fossils and you're like, how would a fish and a frog, what would be it's gonna be something like these things. And we go out and literally find these things. Okay, so that's a little maybe fluffy, sorry, but I think it's amazing. This transition is really well documented. I appreciate you taking the time to make all these observations. Let's talk about some of the details. Before we do that, I want to show you this diagram, which I really like. A timeline right here. This is when those fossils that you just saw are distributed in time. So you can see a lot of them co-occur. Some of them are found in the same rocks. Like Panderichthyes doesn't come from Eustonopteron. You find them together. They lived together in those freshwater ecosystems. So all we're doing here is getting an idea of the transition. What I think is really cool, everybody who studies these animals for the most part thinks that Acanthus stega, which has what you would definitely call arms and legs, is a totally aquatic organism. It has a lateral line on its skull, and we think that lateral line continues down its body based on what we see in the skull. So this is a limbed fish that's not on land. Acanthus stega is probably living an aquatic lifestyle, but it's got what you've got a little bit, but not quite. Really cool. So the thing that's important to me is this variable ecologies and timing. You're gonna see me say this to you so much. None of this ever really is like directional, where it's like, oh, finally, I'm gonna evolve into a tetrapod. These are just fishes living their lives, right? And there's still coelacanths and lungfish right next to them the whole time, not doing these things, right? Okay. Another fact I just can't get enough of, it makes me so happy, and I hope it makes you guys upset and happy. This is these fossils. These fossils are known from these rocks. Tiktaalik, 375 or so. Okay, great. This is a fossil from Poland. It is a fossil of a trackway. It is footprints and handprints in the mud. That fossil is this old.
Oh my God.
So even if Tiktaalik really helps us understand the transition, even if Ichthyostega really helps us understand early tetrapods, that species still existed 10 million years after we know somebody was walking around already. I just think that's really special because it's still contained to the same freshwater environments at the end of the Devonian, but it's not like Eusthenopteron is your ancestor or anybody else's. These are just fishes that help us understand what must have happened. When this came out in 2010, there were a lot of people that were like, because it seems really good, right? Well, guess what? We don't have it all. There's a lot more to learn still. So let's talk about this. 1, 4, 2 sets of synapomorphies up there. Those are all synapomorphies that we can see in tetrapodomorph fish evolution as we go from these fishy tetrapodomorphs, here's Eusthenopteron that you've now seen a few times, to something like this, a true tetrapod. Detailed anatomical synapomorphies that are consistent across the phylogeny. They're really, really cool. We're going to go through these now. On this phylogeny, so after Eusthenopteron, which I think you can all agree looks like a fishy fish. That's why I used it earlier. It looks like a lobefish, Sarcopterygian. It looks like a lungfish, kind of. It still has all of its fins. It's in the water. The first synapomorphy that we can see, and again, the power of fossils is showing us the sequence of change, how these things evolved and in what order. That first synapomorphy, second synapomorphy goes there. I got 4, sorry, zoomed in right there, and we'll add the others later. So here's the first one. Between Eusthenopteron and later tetrapodomorphs like Panderichthys and Tiktaalik and us, is the loss of the dorsal fin. That happens really early amongst these fishes. A full loss of that dorsal fin. So Osteichthyes and Gnathosomes, that common ancestor evolves the dorsal fin. In our lineage, somewhere in here, we lose the dorsal fin. Tiktaalik is very famous for that second character, so it's another tick up the tree. That second character, so not for Panderichthys, but yes for Tiktaalik, what we would call a neck. Tiktaalik got a lot of press, and there's a book about it called Your Inner Fish, you guys might have heard about, a PBS special all about Tiktaalik. Because Tiktaalik has the gill skeleton, the shoulder girdle skeleton, separated from the head. And that's the first time we see this. You guys have a neck, amphibians and mammals and reptiles and birds, everybody's got a neck. Tiktaalik is the first fish to separate this skeleton from that skeleton. So you guys still have one dermal bone in your shoulder, your clavicle. Tiktaalik is the first of these fishes with a little neck. There's other features, of course, if we get into a lot of detail, but I'm just giving you guys some big ones. If we go towards Acanthostega and Ichthyostega, there were 4 things that Acanthostega and Ichthyostega have that you and I also inherited after Tiktaalik. Tiktaalik does not have. Zygapophyses, which is a crazy word. It's written down on your slide. We're going to talk about vertebrae and zygapophyses in lab next week. Zygapophyses are the little teeny bits of your vertebrae that stick off and articulate with one another. So your vertebrae have like a spool shape, like a spool of thread. That's like when you have a disc in your back, most of your vertebrae are those little discs. On a shark, it's just disc, 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 disc. For tetrapods who have to be supporting themselves up on land, parts of their vertebrae, and again, we'll talk about it in lab, articulate with one another. Your back can be rigid and hold your weight against gravity. So zygapophyses, articulations between the vertebrae that are like this, like side to side, help support the body. Acanthostega has teeny tiny little zygapophyses, and you absolutely have zygapophyses. We lose the fin rays, the little rays that a fish has on the end of its fin. Coelacanths have them, lungfish has them, Tiktaalik has them. Tiktaalik has a little elbow, but it still has a little fish fin at the end of its hand. Acanthostega does not have a little fish fin. It does, disturbingly, have 8 fingers, but there are still digits. So digits are present in Acanthostega, Ichthyostega, and all of us. The pelvic girdle. So the shoulder girdle, the pectoral girdle, and the arms, the pectoral fins, there's a lot going on there. We can see that in Tiktaalik and Panderichthys and earlier tetrapodomorphs. But it's only in Acanthostega, Ichthyostega, and of course you guys have inherited this, that there's a full-on pelvic girdle. The endochondral bones that the pelvic fins were each attaching to in a lot of fish is like floating on the side of their body. In these animals, there's a symphysis. Those 2 pelvic girdle sides, the left and the right, meet in the middle. and they fuse, and you have what you fully call a pelvis. You guys have a symphysis in the front of your body that is inherited from that. Also structural, support to put heavy muscles against, to put limbs against and operate off of. What is really cool is that Acanthostega has a pelvic girdle, meaning there's a fusion between the pelvic bones on the bottom of its body on the midline, but they're still floating. The bones are still floating by themselves. In Ichthyostega, oh wait, no, maybe Acanthostega does have it, I'm remembering it wrong. In both of these animals, not only is the pubis and the ischium, these bones in the pelvis, meeting in the front and fusing, but they fuse to the vertebrae. You have a sacrum. Your vertebrae is fused with your pelvic girdle. That is the thing that Ichthyostega has and Acanthostega has. Something's tickling me in my brain. I think maybe Acanthostega is different. But anyway, that pelvic girdle fusion to the backbone, finally. So in all those fishes we've met that have a pelvic fin, it's still floating down there. It doesn't articulate with their spine. So here's how those characters will go on there. Loss of a dorsal fin, separation of the pectoral girdle from the head, and then all these three, zygapophyses, pelvic girdle stuff, losing fin rays on your fingers, unite these fishes. It's very cool. And it's also starting to become uncomfortable to use the word fishes when you look at like Ichthyostega. Something that I think is very nice, well, we'll talk about it in a second. If you want to know the other two characters, we have to go past Ichthyostega into the next period, the Carboniferous. So if I throw on a true tetrapod, like this monster called Eriops, which you guys will meet more next week, this big amphibian, that's where you guys can see these two other characters that helps us complete the transition to tetrapods. And they would go right there. And here's Eriops, I'll you take it in. A big predator from the Carboniferous and the Permian period. It has a whole family that goes back into that period. The two characters that finish off our tetrapod common ancestor are reduction of those crazy digits. Instead of 8 fingers and 7 toes or whatever, the ancestral condition for tetrapods is 5 digits on the hands and 5 digits on the feet. You guys still have 5 and 5 is what we say. 5 up here, 5 down there. So true tetrapods got 5 and 5. And then we lose our last fin. Adult amphibians and adult other tetrapods all do not have tail fins, except for certain cases. So what I think is fun about losing the fins, you can think about it in the tetrapod evolution scenario you guys just saw. Dorsal fin goes first, and then you lose the fin rays on what are your hands and your feet, and then you lose your caudal fin or your tail fin. I think there's something kind of nice to that because you guys learned a caudal fin, a tail fin is a synapomorphy for all of chordates, not even vertebrates. And so everybody we've met that's a chordate so far has a tail fin until we go up into tetrapods and they finally lose that tail fin, which I think is very cool. It's a very ancient thing. So that's how those features map. And again, what I want you guys to really take in and think about is how these changes were not directional. I love this piece of paleo art. This is a piece of paleo art showing one of these flooded freshwater forests. This is a fossil assemblage from Brazil. And so all of these animals are found together in that one place, in that one time. You can see little tiny actinopterygians. You can see early interesting amphibians with cool gills, and then some tetrapodomorphy guys and a lungfish there in the back. The point is these habitats are full of these animals. Only some of them are doing the things that you would say make them tetrapody. Fleshy fins that can move you around. Breathing air with a lung thing off of your gut. These are ancient features that totally predate anybody thinking about going onto land. And in these habitats, these happen. Almost everybody's a carnivore, which I think is very cool. What I would have you guys think about is what are the selective pressures that might allow or encourage evolution to have one or two of these lineages start to become more terrestrial. There's a lot of really good ideas. These are hard to test, but we can look at the modern world to get a sense of what we can believe, what we can think is likely. We already know that up on land, it's 1 million plants and all the bugs you want, and nobody's eating bugs that isn't themselves a bug up on land. So there's obviously a lot of food up on land, interesting. A place to lay eggs. There's fishes today that leave the water they're in to go lay their eggs in like a backwater or a channel that's off the main river. So going for reproductive purposes is a pretty strong selective pressure. And if you had a couple lineages do that, that would be something. An escape route. These are freshwater ecosystems. Climate always changes, seasons always happen. Maybe there's places in these habitats on Earth where ponds dry up and it will be adapted to be able to get out of your water hole if your water hole is drying up. These are all ideas and they're not mutually exclusive, but they're really fun for you guys to consider. You can think about it this weekend. The last thing I'll leave you with is this. Here are three different, completely unrelated actinopterygian ray fin fishes that right now, if you had binoculars, but you don't need binoculars, you can go out into the world and watch, climb up onto land for all kinds of different reasons. You guys have probably seen mud skippers. So these are actinopterygian ray fin fishes that come up onto the mud flats. They breathe air with their little lung thing, and they display sexually to each other. The males stake out territories. It's almost like they're seals, but they're fishes, and they live on land all the time. This catfish and this fish, which is called a perch, it's not related to our perches, they both locomote on land to travel to different water sources. Like they switch which river they're in. They can just get up. This one slithers kind of like a snake. This one is a straight up normal looking fish, and it uses its gill cover, which has spikes on it, to go and pull itself with its gill covers. So not weird today to have totally fish leave the water for different reproductive, survival, who knows, selective reasons. So it's really fun to imagine how little it would take if you have a world covered in flooded swamps with these kind of animals in it, one or two of them to be like, I'm gonna stay up here, probably. Okay, I'll leave you with that, and I will see you guys next week.
Uh,